So many thanks for this opportunity to give this presentation on this membrane protein workshop organized by LINXS. I will talk about aquaporins, my dear friends, since many years, and the focus here is their expression, purification, and characterization. So what I will tell you about is, I will summarize a review article that I wrote together with Roslyn Bill, and this was very recently published, in fact, last week in BBA Biomembranes. So the whole idea that Ross and I had was to look into the high resolution structures, exactly as Ross just described. These are good indicators of successful protein production. So we would like to see what we could dig out by looking at the process for their production and purification to prepare material for both functional and structural characterization. And the hope is, of course, to find a common theme that we can learn from and use for novel targets, especially then aquaporin targets. I would like to start with some inspiration, though. Ross was a postdoc when I was a PhD student in the same lab, and we both struggled with membrane protein production, the well-recognized bottleneck for structural characterization. 20 years back, we had this review from Tate and Grishammer. I saw that Tate is also in the audience. Overexpression of integral membrane proteins for structure studies. And back then, we had 6 structures. And looking now at the graph from Stephen White's homepage, is fantastic to see the development and the increase of membrane protein structures. The focus now is aquaporin work. And worth noting then is that we had the first aquaporin structure year 2000, and this was the human aquaporin 1 derived from red blood cells. It was determined to 3.8 angstrom resolution by electron diffraction. So for those of you that are not so familiar with aquaporins, I would like to start with a very brief introduction to the protein family. This is in their monomeric form. They are 30 kilodalton proteins, and they share this topology of 6 transmembrane helices. They have 2 highly conserved NPA motifs, one in loop B and one in loop E, and they form each of them half helices that are bent back into the membrane, and where they meet, they form the water filter and the proton exclusion. So that is a selectivity filter of the channel. There is also further towards the entrance, one additional filter that is called a specificity filter. And that is, that specific region determines whether only water will pass through or also larger, more hydrophobic molecules like glycerol. The functional unit of the protein is a tetramer, and there we have 4 individual water channels. Worth noting also is that we have cytosolic N terminus and C terminus, and these are highly interesting entities of the protein since they commonly provide binding sites for regulatory proteins, and this is very much the focus of the aquaporin studies now. So here they are, the 31 aquaporin structures that we know of today. So on this list, we have all aquaporin structures below 4 angstrom resolution as collected from Stephen White's homepage. And what we see here is in fact 20 years of development in protein production and structural characterization. We have 31 structures in total, and that they are coming from 15 unique proteins. So in many cases, there are more than one structure for a certain target, and this commonly goes together with an increased resolution over time. We can also see that we have an absolute dominance of X-ray structures, but quite a good fraction of ED structure, and notably 1 NMR structure. In this table, we then have the origin of the target the source for the production, the host, in the case we have recombinant production, the method for structural evaluation, the resolution, the PDB code, and the reference. The white area that you see here, that are proteins coming from the native source, while the yellow area below, that is proteins that are recombinantly produced but using the homologous host. And here we have the bacterial isoforms. The gray area, that is recombinant production, but it's using heterologous host. And that is what we commonly regard as the very challenging process that we want to learn and handle better. Looking at the origin of the aquaporins, we can conclude that they are coming from all kingdoms of life. We have a good representation of eukaryotic proteins, that's the white bars. And here we have 24 in total, 14 from mammal, 17 from mammal, 4 from plant, 1 from a eukaryotic microorganism, 2 from yeast, and then we have 7 prokaryotic proteins. And here, most of those are from E. coli. So here we see the production of structures for aquaporins per year since 2000. We can then see how we, in the beginning, had the majority of our proteins produced from the native source or homologous recombinant production, and that are our bacterial isoforms. But with time, we see more and more examples of heterologous recombinant production. And in fact, from 2014, the heterologous production is dominating. And if we look into our figures, what do we learn? We can see that the majority of the structures are derived from mammals, and nearly 60% of those are recombinantly produced. If we then focus a bit on the human isoforms, which we very much do, since we have many links to diseases and a high interest in studying and understanding those. So the 13 human isoforms, we have 6 of those represented by 8 structures. And in fact, the human isoforms thereby is the most common source of all aquaporin structures. Focusing a bit on the eukaryotic aquaporins, we see that the majority of those are recombinantly produced. If we then look at the host used, we see that yeast, that is the yellow part here, is a dominating host system. And in fact, Pichia pastoris is or absolutely the most common system. There is one structure coming from production in Saccharomyces, that is human aquaporin 10. We have one successful example of producing aquaporins in E. coli, that is the Plasmodium falciparum aquaporin, the malaria aquaporin. And then we have insect cells, as we just heard of, that is not so common for aquaporins, but we have 2 proteins, human aquaporin 1 and rat aquaporin 4, that are produced in that system. Very often, the secret behind a successful recombinant production is connected to the genetic design and the protein design. So we looked into this also for the aquaporins, and we see that we most commonly have a his tag introduced in the protein. Looking at the eukaryotic targets, we see that it could be located either in the N or the C terminus, while looking at the prokaryotic targets is always located in the N terminus and is always connected to a cleavage site, but the cleavage or the protease used differs a bit. Other common tricks to succeed with production and also crystallization is to truncate the hydrophilic extensions. And this is also applied to some extent for the aquaporins, but it's not that common. Another optimization process is the codon optimization to get a more efficient transcription event. And this is also used for the aquaporins to some extent, but not in all cases. Looking at the purification, we have one specific step for the aquaporins that is a bit untypical, and that is a membrane wash. So after the membrane isolation, it's very common to include a wash by urea and sodium hydroxide. And the idea with this is to enrich the yield of integral membrane proteins and then also get a higher efficiency in the solubilization step. But this, of course, requires that the protein is stable enough and so that it doesn't lose fold and activity upon this treatment. Looking at solubilization, we see how we have OG as our dominating detergent. NG is used to some extent, and we see exactly the same pattern when we look at the purification buffers and also the buffer used for preparing material for the analysis, the structural analysis. So we conclude that aquaporins are stable in glucopyranosides and that OD is the most common detergent. Looking further into the purification, having the his tag means that we commonly use nickel purification, but ion exchange is also used for native proteins, of course, but we also have quite some examples of very high production yields, and then we can work without any affinity tags. The second purification step, size exclusion chromatography, is very common, and the aim is, of course, to get this monodisperse peak as indicator of homogeneous material. The yields are up to 25 milligram per liter or 1.1 milligram per gram cells. So if we reflect a little bit on the purification, so as we've heard here and also from Ross and others, we use these high resolution structures as indicators of successful protein production. And this is very much because we get a good yield. I mean, we need to have a good yield to continue with crystallization efforts or other methods. And it's also a good indicator of stability if we can work with structural analysis. One thing that is a bit surprising is that the yield, even though it's very important that you produce good yields of high quality proteins to get a good progress with your structural analysis, the yield is not always reported in the publications. And in addition to that, there is not a systematic way to report the yield. So the comparisons between different production trials is not straightforward. But for those where a yield is reported, we can see that for a eukaryotic aquaporin, the plant aquaporin, SOPIP21 is the highest reported yield when produced in Pichia pastoris. That is a 25 milligram per liter. If we look at the prokaryotic aquaporin, aquaporin set is produced to good yields in E. coli, 13 milligram per liter. So we have these challenging membrane proteins. We know that we need good hosts to produce them, and we also have to reflect on the genetic design. And what do we then learn looking at all the aquaporin structures that we have? That is that simpler eukaryotic host like yeast has worked well for aquaporin production. And a rather simplistic approach for the construct design commonly works, but that does not, worth noting, exclude that for certain aquaporin homologs, you need to spend quite some effort on specific optimization procedures. So by that, I think we can say and conclude that we have to some extent overcome the hurdle of production for the aquaporins. We have quite a good idea on how to produce them, and we have a consensus on the best way to proceed. Prokaryotic proteins, try E. coli. If you have a eukaryotic isoform, Pichia is a very good choice. Membrane wash is a good idea, and this two-step purification procedure, including the nickel tag, is advised, and we see that crystallization has been the successful way forward, working with OG throughout the process. Looking forward, we will still need access to recombinant material to continue progress within structural biology. But what we would like to see is the inclusion of full-length proteins and also protein complexes, because that will assist the understanding of the function in the cellular context. We also have a great development within ED and NMR, so it will be interesting to see what that will add to the field. Finally, many thanks for listening, and I would like to thank also those that have supported us throughout the years for Aquaporin work, the Swedish Research Council, the Brain Foundation, and Biotechnology and Biological Science Research Council in UK. Thank you so much.
Thank you very much, Kristina. We have several questions. One from Susanna, who asked, why do you think that Aquaporins is such a success story in terms of structural characterization?
Why they are?
Mm-hmm.
I think in comparison to other protein families, they are quite easy to work with. They behave better and they are stable.
From Rajiv, what's the importance of the washing of the membrane, that step when you wash the membrane? What
the importance is, the thing is that you wash away the peripheral proteins and you get just the integral membrane proteins left. So that means that you get an enrichment. So you have more of your material. So it's easier to get a lot out simply. It's like a purification step in a way.
Thank you. From Roslyn, why do you think yeast produces human aquaporins so well?
I can't tell for sure, but I always heard that plant protein. Sorry, once again, the question.
Why do you think yeast produces human aquaporins so well?
Okay. I don't know. I was into plant proteins because that is a good match, but no, I don't know.
Me neither.
Sorry?
I don't know either. I just think it's really interesting.
No, sorry, Ross. Sorry to disappoint you, but it's just a good match.
Like you and me.
Yeah, perfect match then.
We have, what are the most used cell breakage method when you're using here?
Yes, what we learned is that breaking cells with glass beads, that's superior to other techniques. That's a collected experience. So a good device or large device to shake a lot is good to have.
Thank you. I might ask that you said that there were different reports regarding the yield. You mentioned something between 13 and 25. What is your experience? Like, what is that works for you in like as a cutoff?
Okay. So that's also a bit tricky because you grow your yeast cells to very high density. So compare per liter is not. It differs so much between different systems you use, but I think 10 milligram per liter using a PICA system would be good. That simplifies.
Thank you so much for your presentation and also for the answers.